Hello there and welcome to this Biology Mind A-Level tutorial on magnification and resolution. So in our last lesson we learned about how to calculate the actual size of cells, we looked at how a light microscope can be used to determine actual cell size, and that the stage micrometer is a ruler which shows the true length of cells. You now understand that the eyepiece graticule needs to be calibrated with the stage micrometer before measuring the length of cells, and how to calculate the cell size when given the size of the image and its magnification. So in our tutorial today, we're going to be having a look at two key learning objectives. Number one, understanding and calculating magnification, and number two, resolution. And we'll be working through lots of worked examples. So let's look at the meaning of these two words, magnification and resolution. So magnification is the size of the image compared to the real size of the object. So you can see that these are different sizes here. Whereas the resolution is the ability to tell the difference between 2 points. So you can see that the resolution of this image here is worse than the resolution of this image. So magnification is the ability to make the image of a specimen larger than it actually is. So microscopes use specialised glass lenses which alter the light passing through a specimen to magnify its image. The image can be viewed by the observer or can be projected onto a screen. So to calculate magnification, you must know the size of the image and the size of the real object. And there's a really handy equation you can use, which we met in our last tutorial, which is magnification equals size of image over size of real object. And a really useful thing to do is to, if I say size of image is I, size of real object is the actual size A, and magnification is M. A really useful way to do this is to draw a triangle. We know that I is on top because size of image equals magnification times the size of the real object, or as I've written here, magnification equals size of image over the real object size. And therefore this triangle now gives you every way to work out each of these different values. Resolution is a numerical measure of how detailed and clear an image is. It's the ability to distinguish between 2 points which are close. So this is much higher resolution because we can distinguish between these points, whereas this is a lower resolution, we can't distinguish between the points. So now let's work through some examples. So question number 1, a sperm cell measures 10 micrometers in length. Please calculate the magnification of this image. So give the video a pause and have a go, and we'll work through it together afterwards. Okay, I'm going to reveal the answer now. So the magnification is times 9,400. So let's see how we got there. So first of all, you work out the image size. So you measure the size of the image. So put the zeros part here, and we see where the head of the sperm cell comes up to, and it's 9.4 centimeters long. Okay, so we've written 9.4 here. Now we know that the sperm cell actually measures 10 micrometers in length. So we know that the image size is 9.4 centimeters, but the actual size is 10 micrometers. And remember our equation. We can work out the magnification from these two values. However, in order to do that, we need to make them the same units. So to make them the same units, I always write the units on the same side or as each other. And what I would do here is multiply by 10,000, because going from centimeters to micrometers is multiplying by 10,000. Now I know this because I've broken it down in my head. From centimeters to millimeters we multiply by 10, and from millimeters to micrometers we multiply by 1,000. Therefore, from centimeters to micrometers, we multiply by 10,000. So therefore, we do 9.4 times 10,000, which is 94,000 micrometers. And we can now plop our two values here into our equation because they now have the same units. So we know that magnification equals image size. Magnification equals image size over actual size. So magnification equals 94,000 micrometers, the size of our image, over actual size, which is 9,400, which means that that is our magnification. So that is how I worked it out, and see how it's really key there to get those units right. You need to get those units of measurement the same before you plot them into the equation. It wouldn't have worked if I'd done 9.4 divided by 10 micrometers. We would have got the wrong answer there. I need to make sure that I do 94,000 micrometers divided by 10 micrometers. Okay, so really important to get those units the same on the top and the bottom of the equation. So that's the first one done. So that is our magnification 9400. Always show you're working, because even if you do it wrong, you'll get marks for the process. Now, let's have a go at the second question. So have a pause of the video and we'll go through it again, just like we did in the last one. Okay, I'm going to reveal the answer. So the question was, calculate the actual size of the nucleus of this animal cell in micrometers. It has 100 times magnification. So the answer is 290 micrometers. How did we get there? So first of all, we need to work out the size of the image. So it says, remember to read the question really carefully. It's not asking for the size of the cell, it's asking for the size of the nucleus, and this is the nucleus. Now it runs between this point and this point, which reads out at 29 millimeters. Notice that unit there, millimeters. The question wants our answer in micrometers. So we need to convert our millimeters to micrometers. Now in the last question, I told you that from millimeters to micrometers, we're multiplying by 1,000. How do I know that? It's because there's 1,000 micrometers in 1 millimeter. So we can now multiply 29 millimeters by 1,000. Sorry, that should say millimeters there, not centimeters. And that gives you the value of 29,000 micrometers, as you can see here. So that's the size of our nucleus in the image. It's 29,000 micrometers, or 29 millimeters. Now that we've got our units the same, so our question wants our answer in units, we've now got our image size in units. We can now plop it into the equation, so we know actual size equals image size divided by magnification. So actual size is going to be image size, which is 29,000 micrometers, divided by magnification, which is 290 micrometers. And I know that because I've cancelled out those zeros there. So that is our answer right there. So well done if you got that right, and don't worry if you don't, we're going to have another go now. So calculate the actual length of this nerve cell in micrometers. Notice that unit there. Give your answer in standard form. Okay. And note this key here, so it's telling us that 2 centimeters in the image size is equal to 4 micrometers. So give this a pause now and have a go at working this out yourself, and I'll reveal the answer in a sec. Okay, so I'm going to reveal the answer. So this is our answer, 1.84 multiplied by 10 to the 4 micrometers. Now, let's work this out. So we first of all work out the image length of the blue line. So I said it's 2 centimeters across, which is equal to 20 millimeters. We then convert this into these units. So from millimeters to micrometers, we're multiplying by 1,000, just like in the last one. So we're doing 20 times by 1,000, which is 20,000 micrometers. Now, what we're going to do is we're going to plop it into the equation. So we do 20,000 micrometers divided by 4, so the magnification is 5 times. So what we do now is we do 20, which is our image length, divided by 4, which is our actual size, to get our magnification, which is 5 times. Now we need to work out the image length of the nerve cell. So again, just like what we did in our last question, we need to measure from here to here, so from the end of the nerve cell to the other end. And that reads out at about 92 millimeters. Now we need to again convert that to micrometers, because that's what our question is asking for. So we multiply that by 1,000 and we get 92,000 micrometers. Now, we already calculated on the last slide that our magnification is 5 times. So I'm just going to flick back there just to make it really, really clear how we got that. We know the magnification is times 5 because we plopped it into this equation here, and we knew that the image size here of 20 millimeters divided by 4 equals 5. So it's times 5. Now, we can then use this equation again with our magnification now that we got from that little key that was given here, and put it into this equation to now calculate the actual length of the nerve cell. So we've got our magnification here, and we've got our image length here of our nerve cell. So we're going to do 92,000 divided by 5, which is going to give us 18,400 micrometers. 18,400 micrometers. And we need to put that into standard form. So to do that, we're going to flip this decimal point back here. It's moved 4 places, 1, 2, 3, 4. Therefore, I know that that is equal to 1.84 times 10 to the 4 micrometers in standard form. So remember to read your question really, really carefully. The units really matter. The fact that it's in standard form really matters as well, because you're not going to get all the marks if you give it in this form. You need to give it in this form. Plus, if I just flip back to this slide, it's really important to use all the information given to you. So don't be caught out. The reason why I stumbled over my words earlier is because I was briefly caught out because I forgot that we'd got this information and that we could work out our magnification. If I just pop back onto this slide, we could work out our magnification from this here. So that's our tutorial complete. Well done for following through. I hope those worked examples made sense. Go back and do them again if they didn't quite, and hopefully with my explanations that will all make much better sense. Well done, and I'll see you for the next tutorial.